In the first part of the talk, I'm really going to talk about some of the genetics behind Alpha-1, which I'm sure a lot of you in the audience knows. So it's going to serve as kind of a review for that. And then I'm going to get into this thing called epigenetics that Sandy just talked about, and I'll let you know what that's like too. So I'm going to start with this basic question, which is, why are we all here today? And the answer, of course, is you should blame your parents. We all inherit half of our genes from our mom and half of our genes from our dad. And Alpha-1 is a genetic-based disease. So we can blame our parents. So what does the genetics look like? Again, this is probably a review for many of you. This is what the Alpha-1 gene looks like in the human genome. Every single gene in our body is encoded by things called exons and introns. This is the DNA code, so if we were going to zoom into these spots, we'd see AGCT or some variation of that. If we have a normal copy of Alpha-1, this gene is going to go through this process called DNA transcription. I promise this is not going to be all a big science lesson, I promise. We go through this process called transcription to make this mRNA molecule. Then we go through this process called translation. It makes protein. This is a normal-looking Alpha-1 protein. This is what we hopefully would all like to have. If this protein is normal and functioning in our bodies, we feel great. We don't have any issues. This is what we would love to have. Unfortunately, with Alpha-1, we don't, Alpha-1 deficiency, we don't see that. So the healthy gene we refer to, or people who have two copies of a healthy gene, we refer to them as PIMM. Why two M's? Because again, we get one from mom and one from dad. This genetic programming, if we have this, we're benign. We don't feel any problems. However, we know through lots of research that there's lots of mutations that happen in this gene. And of course, the most common one that I'm sure, again, a lot of you know about is the Z allele, or PiZZ. People who have two copies of this allele, the Z allele, both one from mom and one from dad, are the ones that are at the really high risk of getting AATD. If you were MZ, you have a much lower risk of getting AATD. There's a few other mutations that we know about. One of them is called S. This is also a pathogenic allele or mutation that can occur in this gene, but it really needs to be in combination with that Z allele to really give us risk for Alpha-1 deficiency. If you're getting both S from mom and dad, you have lower risk. So what does this mean, right? So if we have these Z alleles or S alleles, what are we really talking about here? Well, it means that we have a genetic code for Alpha-1 gene that has these mutations in it. Those mutations lead to a strange-looking mRNA. When that mRNA gets translated into the Alpha-1 protein, it doesn't look normal, meaning that it can't function normally in our bodies, and because it doesn't do what it's supposed to do, then we lead to all of these problems that leads to the AATD syndrome. So this is really the underlying problem that we're all facing with AATD. And the way that we, and another way we can get to AATD is to have what we now also call null alleles. And this is where you have so many mutations in this gene that you don't even get to mRNA production, or you get to mRNA, but again, you don't have any ability to make protein. So essentially, again, the same problem. You don't have normal protein in your body. So the way that we try to deal with this, of course, is to give back normal copies of AAT. So if somebody doesn't have a normal copy of the protein, essentially we take the normal protein, put it into your bodies, and then hopefully the symptoms that you feel with AATD are ameliorated. That's essentially where we are today. This is essentially what we can do with, or what we understand about the genetics. So I've got two things in my title, genetics and epigenetics, so is that the end of the story? No, not quite. And it's because we also know that even on therapy, lots of patients have different responses to therapy. And of course, the way that AATD looks in patients is different. And so even though we understand a lot about the genetics underlying this disease, there's a lot of stuff that the genetics can't quite explain. And this is where this new field of epigenetics comes in. So we can ask the question, why is there a variable response to therapy? And that's really where this new science comes in. So over the next few minutes, I'm going to tell you about what epigenetics is. And I'll bring it back to Alpha-1, I promise. So epigenetics sort of takes the idea of genes and genes determining what happens to us and sort of turns it on its head. It's sort of the truth or our understanding of what we all used to hear from our moms, you are what you eat. And epigenetics is studying that at a molecular level. So it turns out you actually can't blame everything on mom and dad. Some of the things that we experience in our environment also affect the course of the ATD. So to begin describing epigenetics, I'd like to start with this question. What makes us unique? And the pictures here are two identical twins. And identical twins are identical. They look the same because they have the exact same genome, the exact same genes, right? And so if one of these individuals had ATD because they had two Z alleles, their twin is also going to have those two same Z alleles, right? That's what makes them identical. We can ask a very interesting question, which is, if you put these both individuals on supplementation therapy, would they respond the exact same way? And the answer is often no. So that's interesting. Why? So does everybody remember this Human Genome Project that the National Institutes of Health started? I'm getting a couple of nods in the audience. The idea behind this in the NIH was that if we understood what the human genome looks like, this is the whole set of every single one of our genes, then we're going to be able to solve all human disease problems. Because the idea back then was that from a molecular biologist point of view, who you are is entirely determined by your genes, or almost entirely determined by your genes. So the NIH spent decades, millions and millions of dollars. Craig Venter, this guy here, started a company to aid the NIH or compete, depending on your point of view, to crack the human genome. And they did it. And the end result was that we, as scientists, had to come to terms with the idea that actually you are not just a product of your genes. And this was really upsetting to us because for a long time, we've really bought into the idea that Watson and Crick discovered everything you need to know about clinical science. That when the DNA molecule was discovered, that's really dominated the way that we think about clinical medicine and clinical research. And what we now know is that DNA is not your destiny. There's a huge environmental component that can shape what your disease looks like. I'm not saying that genes don't matter. I want to be really clear about this. Obviously, Alpha-1 deficiency is a genetic disease. But the way that that disease plays out throughout your life is going to be affected by environmental things. And one of the huge examples of this, of course, is smoking. Right? Smoking is not something that is encoded in your genetics. It's something that you experience in your environment. So it's sort of, again, playing with this idea, who raises the baby? Is it nature or is it nurture? And so the underlying idea here is that if it truly was all about your genes, and again, we can think about identical twins, then the way that identical twins are made up at a molecular level shouldn't be affected by environment. So you should really have the same health outcomes in identical twins regardless of age or experience, regardless of what they experience in the environment. So people looked at this. They took a whole bunch of identical twins, what we call monozygotic twins, and they examined them at different ages of development. And the bottom line here is if you look at their genetics, if you look at 3-year-old twins or people who are over 50, their genetics are going to be the same throughout their entire life because they are identical twins. But look what happens when you now start looking at what we call epigenetic differences. The identical twins who are young look pretty much identical. You can find almost no differences in an epigenetic level. But when you now look at 50-year-old twins, you find lots of differences. So the question of what makes those 50-year-old twins unique, why are they not the exact same people, why do they, more importantly for us, not have the same outcomes, health outcomes, it's because probably of these environmental and epigenetic changes. And so this is what we're really excited about. Hopefully we can study more in depth. So we now like to think of epigenetics as part of the missing pieces of how our body works and responds in a disease setting. Another example of this is sort of an old high school biology question, which again, we've only had answers. This whole science is fairly brand new. We've only had it or known about a lot of this stuff for the last decade or so. You can ask the question, how can we get so many different types of cells in our body from one genetic program? So think about this. Every single cell in your body has the same set of genes, right? We get half from our mom and half from our dad. So how is it that we have so many different kinds of cells? We have skin cells, heart cells, liver cells, lung cells, immune cells. They all come from the exact same genetic code. And we didn't really have a great answer for this until we really began to study this thing called epigenetics. Because what epigenetics is doing is it's shaping how the gene program is used. So we can start with a stem cell, which can become every kind of cell in our body. And what happens as that stem cell differentiates into different kinds of cells is this epigenetic programming is actually controlling how our genetic code is used, turning on some genes, turning off other genes. So if you're an immune cell, you want to make sure that you're turning off the nerve cell genes. And if you're a nerve cell, you want to make sure that you're turning off the immune genes. And that process actually occurs through what we now call epigenetics. So how does all this actually work at a molecular level? Again, sorry, little biology lesson here. So this is what our DNA looks like. The DNA in our bodies and every single one of our cells is compacted into a structure which is called a chromosome. If you were to pull the chromosome apart until it's really taut and tight and you couldn't pull it anymore, what you find is this structure called chromatin. And what we refer to this structure as beads on a string. And the string part here is our DNA. So if I was looking at the Alpha-1 gene right here, this is where it's at. It's sitting right here, and there's AGCT on this. These beads are the part that epigenetics controls. And so these beads are actually proteins. And if we're to zoom in on one of these, it kind of looks like this. So the DNA is wrapped around this structure, which is called a nucleosome. And these proteins can actually change through chemical modifications. And one way, or one easy way to start to think about this, is that the DNA, the string, is really, that's the nature part. That's the part that we can't really change. That's the part that you got from your mom and dad. It's these beads and these proteins that can change. That's the nurture part. A really simple way to think about this is that under the genetic model, when we think about any gene, we think about, again with Alpha-1, we think about you can have a normal allele or a normal copy, that's the M. And if it's on, if you have a normal copy, you can think of it as being on. If you have a null allele or a mutated copy of that gene, it's off because you're not making a normal copy of that protein. With epigenetics, we actually turn that on-off switch into more of a dimmer switch. Because what we're now saying is that you not only have the underlying DNA code that you inherited, but you actually have the ability to modulate the level of that expression, the level of how that gene is being used. And that's really what epigenetics has taught us. So here's a good definition. And here's the parts that when we discovered this and truly understood this, it was really mind-blowing in terms of how the implications could potentially affect disease research. So we know that epigenetics really don't have anything to do with the DNA sequence. But one of the really fascinating things is that what's wind up changing these proteins, what's leading to these modifications of these proteins that changes our epigenetics in this dimmer switch is actually the environment. And when we ask the question, what kinds of things in the environment, it's almost anything that we've studied. Again, this is at a really basic level in petri dishes in the lab. But if you think about diet, if you think about exposure to particles that are in the air, even if you think about sort of quality of life stuff like stress levels, all of those things have the potential to change your epigenetics. The other really important thing here is that we now also know that these epigenetic changes, just like our genetics, are inheritable. So maybe we can go back to blaming mom and dad after all. We know that these epigenetic changes, those environmentally induced changes, are actually able to be passed down from one grandparent to a grandchild, and the latest data is really suggesting that it can be as far as a great-grandparent to a great-grandchild. So these changes that occur due to environmental exposures are actually inheritable and can influence how our bodies deal with disease states. So epigenetics, along with genetics, really are sort of the missing pieces that we would love to start to begin to apply towards asking the hard questions about Alpha-1 disease, which is, why do people respond differently? Why does the disease look so different in various people, even if they have very similar genetics? So it's not really nature versus nurture anymore. The way we really think about this now is that we need to look at both. If we're truly going to understand disease in individuals, we need to look at both nature and nurture. So if we think about AATD and we put it on a spectrum of being heavily influenced by our genetics or heavily influenced by our environment, we know that certain really heavily environmentally influenced diseases like asthma sit on this side of the spectrum. We know that genetic diseases like cystic fibrosis are obviously sitting on this spectrum. Our hypothesis right now, although the research is just beginning, is that AATD sits obviously towards the genetic side, but we think there's going to be some environmental contributions to how the disease plays out in an individual. So I'm going to go back to this slide of our Alpha-1 gene. So here we know our mutations that can happen. This is the genetic side. We know that the Z allele or the S allele places a higher risk of getting AATD. So what about epigenetic influence? Well, we know that this gene is actually under some epigenetic control. And I'm not asking you to, or I'm not going to even go through and explain what these are, except to show you the complexity of the epigenetic shape underlying this gene. So when we ask what the proteins that I told you about that are associated with this DNA, what they look like, you can see that all of these sort of peaks and valleys, those are all changes in the shape of those proteins that can affect how this gene is used. And this is the epigenetic shape that we really want to begin to study and understand how it contributes to the disease that people see. So we actually do have a study ongoing at NGH right now where we are doing this. So the NIH funded a study that is called Epigenetic Regulation of Immunity and Alpha-1 Antitrypsin Deficiency. We have a number of goals, but the bottom line here is that we don't have a lot of information about how epigenetics is contributing to AATD. And this is really one of the first studies where we're going about trying to understand how these epigenetic mechanisms are affecting the disease state in an individual. We've begun to do this, and we've already found differences in a number of genes that we know are involved in the immune response in AATD. We can find changes in some of those protein shapes and the epigenetic profiles for Alpha-1, for IL-8 and TNF-alpha, which are inflammatory genes that we know are involved in some of the lung issues that you see with AATD. And what's also encouraging or exciting in the future is the possibility of using some of these epigenetic mechanisms to actually affect the level of expression of Alpha-1. So we can use epigenetic modifiers and actually already available epigenetic-based drugs to affect how the Alpha-1 gene is used in our bodies. So none of this, of course, would be possible without technological improvements, and that's sort of the story with everything that we face today. So I said earlier about the Human Genome Project that it took the NIH in combination with Craig Venter decades and millions of dollars to do one human genome. Today, it's amazing what we have available to us. We have really literally gone from decades to do one human genome to literally a couple of days now to do one human genome. It used to take a building's worth of equipment to do one human genome, working again for decades. Now we can do it on a benchtop instrument that looks like this. So really what we want to do is take this technological improvements and apply it to AATD and read not just your underlying genetics, but also begin to look at some of these epigenetic changes. And in a way, hope to personalize those diagnostics. If you think about the example I used of the identical twins, that's sort of the ultimate example of personalized medicine or personalized diagnostics. We would love to understand how disease differs in individuals and why it does. And we really truly believe that epigenetics is going to be a big part of that. So just again to plug the study that we have ongoing, we really truly hope that at the end of this NIH study, we're going to have some potential biomarkers at an epigenetic level that will help us identify why or how treatment is effective in some patients and not in others. Our theory is that there's already some underlying epigenetic programming in some individuals that makes them less able to respond to treatment than others. But that's a theory. Stay tuned, we'll tell you in a few years. So with that, I really do need to acknowledge a whole bunch of people that are part of this. So part of the NIH study that I told you about is Sandy, Nabil, who spoke earlier, and Lisa Meyer. We're all working at NJH. Of course, the NIH for funding the study and some other sources of funding for that. This is the study we are actively recruiting if you're interested in being part of this. Our coordinator is Brianna Barks. At the Alpha-1 desk outside, there's flyers and some cards for Bree if you're interested or just have some further questions. You can contact Brianna. So I warned you in the beginning that I'm a little different, so this wasn't a clinical talk about Alpha-1. But I hope that I've given you some idea of where we're going in the future, about what the potentials are for coming up with entirely new ways of thinking about Alpha-1 and hopefully some new types of treatments that are going to be based on this epigenetics and really get us to move beyond just the idea that whatever genetics you've inherited is really going to determine your disease, that there's an environmental component, which I'm sure a lot of you or all of you know at a gut level affects how your disease presents in your daily lives. So thanks for your attention. I hope it wasn't too much, and I'll be happy to take some questions.
So with the genetic research going on, I know there's been a lot of genetic research with breast cancer, and they now have the BRCA testing.
Yep.
Is this going down a path like that? I mean, I know we have Alpha-1 testing,
Yeah.
but where you can predict the likelihood?
Yeah, I guess I would say that it's interesting that you bring up the breast cancer examples, because I think we're at a little bit of a turning point in the way that we think about genetic testing and genetic counseling. And I don't want to make any hard statements here, but I think that we're stepping back from the idea that even with the BRCA1 test, that you want to tell a woman you have this type of genetic background, therefore you're 100% going to get breast cancer. So let's start thinking about invasive treatments. I think there's a growing evidence now that that's not true. And the answer, or again, the theory, although we don't have a lot of evidence quite yet, is that it's really these epigenetic mechanisms laying on top of the underlying genetics that is really, at the end of the day, going to determine what kind of disease you get, whether that's 5 years down the line, 10 years down the line, 20 years down the line, et cetera. The problem is we're not very good yet, or we just haven't had as much experience looking at the epigenetic data and understanding what it truly means. So I think with Alpha-1, that's exactly where we are. This study that we've started is literally the first of its kind, where we are saying, you know what, we have enough clinical experience to know that people who have Z alleles don't present with the same kind of disease. So the answer is why. The question is why. And we hope that the answer is epigenetics. We hope that by examining individual patient samples and looking at their epigenetics, that we'll see sort of those needles in a haystack of saying, okay, yeah, patients who look like this at an epigenetic level, even if they have a Z allele, we think they're not going to respond well to therapy, whereas these other ones will. And then hopefully we can do something about that. That might be 10 years down the line, 15 years down the line.
No, I just was more interested in that.
Yeah, no, absolutely. So yeah, I think the really exciting thing is that if you had asked any clinical scientist, myself included, 10 years ago, was it all about genes? we would have told you yes. In the last 10 years, we've begun to understand how important epigenetics is, and we're just at the start of it. Yeah. Okay. Oh.
Last question, real quick. Decades to days and on the benchtop, what is the prospect of getting that down to hours? What's preventing that?
So the biggest jump that got us to where we are today, and the analogy I use is when the NIH did it decades ago, the technology we had was essentially to say if everybody in the room was a genomic scientist, what the NIH did was give everybody a copy of an encyclopedia. Let's say the encyclopedia was a genome. And we told everybody, go home, read the entire encyclopedia cover to cover. When you understand every single letter and you understand the context of every single word and you know everything that's in the encyclopedia, come back to us, we'll all get in a room and make sure that we're all on the same page. That's why it took 20 years. Now that we have that underlying map, now what we do, and this is what's called next-gen sequencing, is we take that encyclopedia, rip it up into 3 pages. We give everybody in the room a different 3 pages. Say, go home, read those 3 pages. When you understand that, just those 3 pages, come back, tell us what it's about. That was the huge leap. That's how we got from decades to it only taking a couple of days. We are making incremental changes that are getting us faster and faster. About 5 years ago, when I got to National Jewish Health, we had an instrument that wasn't a benchtop. It took up about half of a room. That could do a genome in about 2 weeks. Today, we're down to about 2 days. So I think we're going to get there, and the cost is dropping as well. But we're not going to make the huge leaps that we did before. But there are incremental improvements that are happening. So maybe another 10 years, maybe another 5 years will be where it's at the level of a couple hours. But even today, if you think about a clinical test, we're not that far away. We can do a genome in about 2 days. The cost is a couple of $1,000s. It's certainly not going to be something that we apply to everybody, but if for those tough cases that we really don't understand what's happening with somebody, it's not out of the realm of possibility to do it. Yep. Yeah, so if I'm understanding the question correctly, what's the possibility of having an MZ genotype and a hemochromatosis as well? Honestly, I don't know, and maybe some of the MDs might know, but I think it's just
hemochromatosis is much more common than Alpha-1 and is also a genetically mediated disease that has liver disease as a major component and in its severe form. And it's always been something that we've wondered about, whether they might have synergistic effects, for instance, on the liver, make it more likely someone gets liver disease if they have both of those genes that kind of tend towards liver injury. Although the mechanisms of liver injury are different in the two, we still know that there's, Alpha-1 makes you more susceptible to things that normally injure the liver. So that's a long way of saying that's a great question, and we don't have the answer yet.
Yeah, sorry. I wish we knew more, but at this point we're. Mm-hmm. Oh, okay. I think anytime you have genetic factors that work in different ways, obviously you're at risk for some sort of synergy that unfortunately makes the outcome worse. Going back to the light switch versus the dimmer switch, doesn't that mean then that the genome or the genetic background is a lot easier to study than the epigenome? And the answer is absolutely, absolutely. So our DNA code is made up of 4 bases, AGCT. To date, we're still counting. The epigenome has a potential for over 50 different kinds of combinations. The only reason why I'm up here today telling you about any of this is, again, because of those technological advancements. If this was 10 years ago, we literally couldn't study the epigenome. But today, because the cost has dropped so much, because we can do these assays so much quicker, essentially using the same technology, we can read through the epigenome at a fast enough rate and cheaply enough that we can actually begin to study it. Uh, well, it's definitely not infinite, but the actual boundaries of where it is, we don't know yet. Literally, at least twice a year, somebody is discovering some new kind of epigenetic modification that we didn't know about. Again, the science is fairly brand new for the way that we think about fields of science, and our ability to look at it is also fairly new. So it is complex, but because it's sort of location-based in a way, because we know that the genetics doesn't sort of exist on its own and the epigenetics doesn't exist on its own, the two really are always going hand in hand, that if we want to study Alpha-1, which we do, obviously, then we can just have, we in a way can just target that gene area and the things that we know regulate that genetics. So we don't need to exactly look at everything. We can focus in on the parts of the epigenome that we think are important. That's a great question. So the question is the study just for ZZs or is it for carriers as well? We are primarily interested in ZZs. And in fact, we're most interested in ZZs who have not yet begun therapy. However, as the study moves on, we might begin to look at carriers as well. But at this point, we're most interested in ZZs.
It's a very difficult study to do because the goal of the study in its most perfect form is to look at ZZ patients before they've started therapy and then redo the analysis after they've been on therapy for 6 months to see if therapy has changed those epigenetic markers. And you can imagine someone wanting to put off therapy until they can have their bronchoscopy done, and then coming back 6 months later and letting us stick that tube down their lungs again. So it shows true dedication, but many of our Alpha-1 patients are very dedicated to finding the answers to these questions. Yes, sir. I don't want to step on Brian's toes, but the fact is that we go to the ZZ patients because they're likely to have the most dramatic signal. You know, that we go where the richest likelihood of data is. And then once we identify those things, we can see, do these apply to MZs, to SZs, to MSs, things like that, because we go where the money is first, money being knowledge and information, and then we try to spread that knowledge around where the signal might not be as strong, but it might be playing a big role.
One of the underlying sort of the way that we're thinking about this, and we've borrowed this way of thinking from other diseases, cancer being a big one, asthma being a big one, where they have looked at epigenetics, is that at this point we understand that the disease process itself is sort of one of those environmental signals. So it's sort of like circular logic, but so for patients who have ZZ, we understand that their disease is going to be much more severe than MZs. And so we want to start with what does the severe case look like, because we think that's where we're going to find the sort of the richest trove for those epigenetic marks that matter the most, and then we'll move on. But however, you know, we're at the very beginning of the study, and we'll see where it goes.